Okay, so the question that we're going to be trying to answer with this last little bit of this subsection is how, in many cells, when the calcium concentrations in the cytosol increase, does that lead to a specific response? Because we have said before, when these second messengers, calcium being one also, when their concentrations increase in the cytosol, you have a specific response by the cell. We saw the example of muscle cells where you get muscle contraction, and there were other examples that we've seen. But how does that work? Like, what is one of the mechanisms by which increasing calcium concentrations will lead to a response by the cell? So one of those players in the calcium response is CAM kinases and calmodulin. And we're going to get to know these proteins a little bit. An increase in cytosolic calcium will lead to specific responses by cells. Responses are cell-specific, and responses can be different within a single cell, local responses. Now, for example, we know that at the AIS of a neuron, if you increase the calcium concentrations, that will lead to a form of adaptation, whereas increasing calcium concentrations in the axonic bulb will lead to the release of the neurotransmitter. In many cases, an increase in cytosolic calcium concentration leads to activation of specific proteins, including enzymes. This activation is frequently mediated by a protein called calmodulin. Calmodulin functions as a multi-purpose intracellular calcium receptor. It can be found as a monomer or as a subunit in a larger protein complex. It will be part of the larger protein complex, meaning it will be part of its quaternary structure. Calmodulin has calcium binding pockets, and upon binding calcium, it will undergo a conformational change. It will change its tertiary structure. Upon activation, it will either bind and specifically activate another protein, or if part of a larger protein complex, will activate the protein complex. So here we have a picture of calmodulin. And you can see it's a rather not very large protein, kind of looks like a dumbbell, and it has 4 binding sites for calcium. Once it has calcium bound, it will go on to bind to other proteins. And this is just showing you a small portion of another protein, not the whole protein. That will lead to the activation of that target protein. Among the many targets calmodulin regulates are enzymes and membrane transport proteins. As one example, calcium calmodulin binds to and activates the plasma membrane calcium pumps that use ATP hydrolysis to pump calcium out of the cell. Thus, whenever concentration of calcium in the cytosol rises, the pump is activated, which helps return the cytosolic calcium levels to resting state levels. This is an example of negative feedback. Also note, the calcium calmodulin activated pump is working in concert with calcium pumps in the ER and the sodium calcium exchanger on the cell membrane. So this is a little bit that we didn't ever talk about, because we couldn't talk about it until now. So what does this look like? We always said that calcium concentrations increase in the cytosol by whatever method, okay? There's something that happens upstream, whether it's a signal transduction pathway of some sort being activated or channels being opened, etc., right? That you had negative feedback on this, right? You had negative feedback coming in from the sodium calcium exchanger and antiporter on the cell membrane. And you also had another one, which was the calcium ATPase, which is a pump on the ER. So this is ER membrane, and this is the cell membrane. Now we have another one. And what is that other one? This is calcium calmodulin activated calcium pump. And this is also on the cell membrane. But this is activated by calcium, and its activation leads to further inhibition on calcium concentrations within the cytosol. So this basically ramps up the calcium removal even more, and that's why you get these concentration spikes of calcium, because as calcium concentrations go up, more of these guys are activated. And as more of these guys are activated, you have more inhibition or more removal capacity of calcium from the cytosol out into the extracellular space. And of course, you have it going into the ER by the calcium ATPases on the ER membrane. calcium calmodulin dependent kinase 2, CAM kinase 2, very important protein. CAM kinase 2 is found in most animal cells and especially enriched in the nervous system. Each CAM kinase 2 has 12 kinase subunits. And remembering a kinase is a protein, an enzyme, that catalyzes the reaction where a phosphate group is added to that protein, to the target protein, using an ATP. Each kinase subunit can be activated by calcium calmodulin, so all 12 kinase subunits individually will be activated by calcium calmodulin, meaning calmodulin that has calcium bound. Once a kinase subunit of CAM kinase 2 is activated by calcium calmodulin, the activated kinase subunit can autophosphorylate itself. Once a subunit is phosphorylated, it will remain active even if calcium calmodulin unbinds due to the lowering of cytosolic calcium concentrations. So autophosphorylation, when we say a protein can autophosphorylate, it means it can phosphorylate itself. So it has kinase activity that can act on itself. The subunits are dephosphorylated, thus inactivated by cellular phosphatases shortly after phosphorylation. The phosphatases are always active, so if you don't have the kinases working, they're always there, and they're going to remove those phosphate groups from the protein. So here we have a picture of CAM kinase 2, and it's showing you one side of it, with 6 of the kinase subunits, and these are the kinase subunits in green right here, 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 and here. And you have this regulatory segment right here being shown. Sometimes these kinase domains will kind of pop out, and if you have calcium calmodulin around, it will wrap itself around this sort of regulatory segment, and that will lead to the phosphorylation of these regulatory segments. So you can see two of these kinase domains have calcium calmodulin bound, and once they were activated, for example right here, they went on to then autophosphorylate themselves. And once calcium calmodulin is gone here, no longer you have calcium calmodulin bound to these regulatory segments, the phosphorylation event stays and these kinase units are active. If the calcium spike frequency is high, more of the subunits are activated over time, thus increasing CAM kinase 2 activity over time. At each calcium spike, kinase subunits are activated by calcium calmodulin and then autophosphorylate themselves. Because the lag before the next calcium spike is short, all the subunits will not be dephosphorylated. Therefore, when the next calcium spike arrives, more subunits are activated, thus increasing the CAM kinase 2 activity. If the calcium spike frequency is low, CAM kinase 2 activity will be low. At each calcium spike, kinase subunits are activated by calcium calmodulin and then autophosphorylate themselves. Because the lag before the next calcium spike is long, all the subunits will be dephosphorylated. You have 12 subunits on a single calmodulin protein. If the frequency of calcium spikes is very high, you're getting calcium spike after calcium spike after calcium spike, where the lag between the spikes is very short, then you don't give enough time for this dephosphorylation event that is being shown here to happen, okay? Meaning these phosphate groups won't be removed. And this is what's being shown here, actually. The phosphate's being removed, and you go back to the state that you were at before, right? So calmodulin binds, you get activation of these kinase subunits, they've autophosphorylate themselves, remain active, you get the next spike coming in immediately, and you get more of these subunits, these domains, these kinase domains activated, and it reaches a point where most of the CAM kinase 2 proteins are now fully active with most of their kinase domains active. The more kinase domains you have active, the more kinase activity you have. That goes back to increasing the Vmax if you have more enzyme, right? In this case, you have more active enzyme, so you increase the Vmax. Whereas if you have spikes, calcium spikes that are of low frequency, then the lag before the next calcium spike comes in, the protein is dephosphorylated, and you go back to the original state of very low or non-existent CAM kinase 2 activity. Okay? This is a mechanism which can lead to wildly different responses which are dependent on the frequency of calcium spikes. So the y-axis here is CAM kinase 2 activity. And if you have low frequency of calcium isolations, you have very little CAM kinase 2 activity as being shown in this graph here, right? And if you have a high frequency of calcium isolations, meaning high frequency of calcium spikes, you're going to end up having very high levels of CAM kinase 2 activity. This thing, every 2 seconds you have a calcium spike, and every 20 seconds, okay? Now, this can lead to wildly different responses for here and here. Even though they're calcium spikes, having low activity of CAM kinase 2 can lead to one response, whereas having high activity of CAM kinase 2 will lead to a completely different response. Sometimes these responses are actually opposites of each other. Okay guys, we'll leave this topic here and continue with the rest of the section later. Thank you.